Hello, thank you very much. Thanks, BGI, to let me share some perspective on research with you. I'm Zeying, and I'm glad to share some perspectives today. Kind of remind again, so if you have any questions, just leave your message by Q&A section. Then I will choose some question at the end to answer directly, or I will leave you some message. So before we talk about multi-omics, I'd like to ask everyone a question. Why do we call omics? What is the difference between the metabolome and metabolomics, or proteome and proteomics? To simplify the understanding, we can say that is the narration, big data of proteome and metabolome. Considering that, if I today actually preparing an on-site workshop for you in a research school in a village, not in a huge city, but in a village. I should prepare at least 50 pieces of cakes, hundreds cups of coffee, and more chocolates. But unfortunately, in this village, the cafe has limited storage. They provide me all they're having, and now they're considering. They're shocked. If I will come back tomorrow again, and if they should actually prepare more cakes and more in stock. But now they only have my coincidence show up. They haven't got more profile from my side, just like some publications 20 years ago. They have a single antibody and ran a WB and claimed that they have some relationships to some certain phenotype. That is the proteome analyzed 20 years ago. But like now, the cafe manager is not satisfied with the data set. She got my per case history in their system and realized that I actually just coincidentally have this huge per case today, but she still doesn't know the reason. Then she tried to get other data from me, maybe the calendar from the research school, and then she find the answer. In the day when the school have on-site training course, someone must not just Mrs. Lu. She will come to purchase more cakes than normal. Now we can understand that the whole story and predict when is the next purchase coming out. That is exactly what we are doing in the medicine research. Example, we are aiming to analyze proper precision medicines by enabling personalized and accurate disease classifications, treatment selection, and clinical decision makings. So researchers are trying to integrate as more data as possible from downstream genome, epigenome, transcriptome, proteome, metabolome, and episome, so environmental. And from the healthy pre-disease and disease group, and to get more data integrated as much as possible, from which data to be further makes precision preventions, diagnosis, treatment, and prognosis realistic. Take an example. Study with multi-omics data build a biological function module is so we can see in the picture D is such a calculated biological function models. So by this data analyzed. And then they successfully trained the data to distinguish certain types of disease development. At the same time, they're giving the supplements to some patients. And then we find a rematching of the personal biological function module, which with this integration, it can provide us with a helpful information of the theoretical foundation for improving our understanding for health and disease. So, however, unlike the upstream omics, I mean the genomics and transcriptomics, so we also always told that is upstream omics, and the downstream omics like proteomics and metabolomics, which is closely related to the phenotype. They yield really complex data that challenging researchers. On one hand, metabolomics and proteomics are more complex compared to genomics and transcriptomics, which is a wider variety of basic structures and an extra types of metabolites and proteins. On the other hand, the mass spectrometry instrument, for example, used for these studies, they generate much larger and much complex data sets, heavily relying on the database for interpretation and analysis. For example, in a typical mass spectrometry analyzing, the total ion chromatogram, each peak represents one or more similar substance. And only by referencing the specific instruments data bank can further analyze to be performed. This entire processes, from wet labs to data analysis, need to be fully integrated. This complexity often causes confusion and difficulty for many researchers. How can we make this proteomics and metabolomics research less taboo? So we need a systematic experimental setup and optimized workflow for that. In Voring, I will begin with wet lab for data generation, dry lab for data analysis, and 2 deep dive case study for multi-omics. The first steps for generating the data is the appropriate instrument and method selection. This is based on the experimental process, purpose, and sample types. I'm making an example here is the build of our BGI lab. We have more than 45 different kinds of instruments. Yes, but it's not some connecting games. The truth is each instrument or specific combination of any of them has specific limitations and strengths regarding to accuracy, dynamic range, resolution, and speed of analysis. We are now, for example, taking advantages for deep screening with high resolution MS for untargeted screening. For example, the Orbitrap Astro or the QE platform or the timsTOF. But considering the speed of analysis, we will still use QTrap, triple cross MS, target panels. Anyway, there will be one most appropriate option for your own demand. But since we already mentioned different kinds of categories, untargeted, targets, and semi-targets. So the main objects of omics approach are to quantify biological molecules and categorize how this changes. So with untargeted methodology, they are in the definition than the most omics big data method. They have a high coverage of molecules combined with statistical and bioinformatics tools for the analysis, but they are quite complex in the analyzing data and demand a really strong database. The opposite is target analysis. It can provide accurate concentration of metabolites, but because of the limitation of current mass spec instruments, they have a limitation of coverage from single target metabolites, 1 or 2, or maximum several hundreds, so 200 or more, not more than 200. Mostly is 550 or some. And then the semi-target is a combination of both. It also have a calibration steps referencing several standards, but they have a much more bigger panel than normal targets, but not that accurate than target methodology. I will introduce in deep dive in each topic to make it more understandable for you. So, introducing the untargeted metabolites. Since the metabolites, what we are talking about, is numerous variety of dynamics concentration and physiochemical characteristics. There is currently no single analytical tool of detection techniques can cover all of them. You can see there are some violated and non-violated hydrophilic to hydrophilic. There are different polarity. There is quite complex. There are too much. So the most critical steps in LC, so we talk LCMS, mass. So it's in this liquid chromatography step, is the selecting of the right column very important. The stationary phase chemistry should match the nature of your ideal analyze. We have concluded 4 different types of column in BGI for different physiochemical groups. For example, the C18 is more suitable for non-polar and medium-polar small molecules, like hormones, like fat-soluble vitamins, free fatty acids, lipids, and so on. And at the same time, if you want to see some amino acids, then you will need to, or some similar strong polar and strong hydrophilic small molecules, then you need to amide their column. Also, for plants or lipids who have also other kinds of columns with specific end capping for the stationary place. Another important steps for untargeted metabolomics is the data bank. So after the LC phase, the compound will be performed with fast scanning mass analyzer operation in well-mass range and needs a powerful bank to compound metric. A process of the data analysis is showing here. For the identifying stage, so you see, we have our library, a non-metabolite A, and which should need to be matched with the data output. And so to say, the data input we talked about and is the analyzing input, but it's the MS instrument is the output. So it needs to match together with your measurement this time. And then this measurement should, for example, like candidate annotation and scoring. For this stage, in BGI, so for this stage, the spectra of each compound should be matched with the database and score how fit they are. In BGI, we have our cell-based library local database. So totally with 10K compounds information are saved. This data bank is strictly aligning with their methods, including gradient change, solution use, the LC column, or energy of EC, and so on. And compare with other databases, the identified rate can reach 60% of the detection. After identifying, the data should be normalized, so well prepared for differential expression analysis. At the end, we can put all these differential expressed metabolites back to the pathway map and discover the change between treatments or disease groups. Was the idea. And now we need to sharply switch to untargeted proteomics. So unlike metabolomics, proteins are less complex in the meaning of diverse of physicochemical characteristics. But so they are all peptides after digestion and consist of 20 kinds of amino acids. But the other side is the difficulties are it needs much more deeper analysis of the data. We have two acquisition strategy in MassMax. They are called DDA, data-dependent acquisition, and DIA, data-independent acquisition. To simplify the understanding, I say now, I'm now in the library, and I want to find a specific book on the bookshelf. I cannot find in the first screen. It's a random, similar as a random screening in DDA, instrumental in DDA. They only record the most abundant signals. So the book is at the end is not found. Yes, I just checked through and I haven't found. Another way is sort those books from A to Z, and then I find the book I need. It's much more accurate and promising that more books can be found with a library build-out. The DIA workflow will mix a library from your original sample. And then we will build up a sample library before running the initial samples. With this, the DIA methodology can reach much more than 10K identified proteins in the general tissues. It's actually 5 times better than DDA can achieve. Another method setup I want to introduce for proteomics is the labeling-based approach. We have a set of label called iTrack or TMT, depending on the sample number set you have. The label is bound with the digest peptide in each of the sample. So you can understand now, 18 samples have 18 colorized set of peptides. They will be mixed together as one and run together into LC-MS. Then the quantity, the comparison of the peptides amount will be directly compared through the amount of the label, so the colored. Yeah. So at the end of the untargeted analysis, I will give a short overview of what I mentioned. There will be all provided as a service at BGI. So we have metabolomics, we have different columns, C18 and MIDA for medical or animal research use, lipidomics, plants, etc. And then for proteomics, we have labeling and label-free. So label-free is, for example, the DIA and DDA I just mentioned, labeling with TMT or iTrack. Another part I haven't mentioned yet is some special samples, like protein modifications. We have flow workflow for phosphorylation, acetylation, and glucosylation and ubiquitination. or some special sample type like LCM or FFPE, or some single cell sample. Going back to another type of analysis, target approach. It actually usually starts with assumptions that selection of group of compounds and targets a specific aspect of biological systems. For example, the energy metabolism pathways, the homocysteine metabolism pathway, or all the neurotransmitters. It has in general covers less than 50 compounds. Semi-targets metabolites is several internal standards can be calculated the combination like the normal target one, but is more widely set up for screening like the untargets. The 2,300 panel at BGI is a representative. It covers 700 of metabolites and 1,600 kinds of lipids. The researchers can get all this quantified and qualified data at once. A short summarize, I'm joking that at BGI Genomics, we can provide different kinds of targets and semi-targets panels. And also, you're welcome to send us your list of wish to us, and we can combine some of them with you. And the second part, I would glad to move to dry lab, the data analysis. In mass-based omics data, raw quantitative data must be pre-processed to enhance quality before statistic analysis, ensuring more accurate and reliable outcomes. For target or semi-target analysis, a normalization steps for calibration curve is needed, and then the calculate concentration. In these two approach, the data processing is still not quite complex, but the untargeted approach, for example, the DIA proteomics, identifying which more than 10,000 types of proteins will need to confirm the instrumental QC, sample QC data normalization, and retention modification several steps before we can use the batch data to compare. For example, at BGI, we also use that, like including overlaying of internal standard and BPC overlapping, PCA score plots, and heat map of QC samples. Oh, you are now maybe so confusing about what is this. Never mind, you may not familiar with this map, which is exactly what I'm going to answer. How could we work with these huge amounts and complex data? Data visualization can be integrated at many stages of the data processing and analyze workflow. It may use for exploratory purpose to gain an understanding of the data's underlying structures and patterns. For example, the PCA, I'm showing here in this left side, is a method of obtaining a few comprehensive variables. So to say, they have reduction of dimension tree. We can use for preliminary observation in the groups between or in the groups or between groups differences. A PCA is unsupervised, means we are not considering grouping information in this stage, which can actually help us to find the abnormal samples. Another side is the PLSDA. It's a similar method, only that it's supervised. We use the grouping information to build a module and manually classify the samples. The classifying is better than PCA and can be used to screen different metabolites. For judgment of the module, it was also calculates the validation plots. Now we are finished with the similarity. We can now see what is the difference in the groups and what is the difference between the groups. Then, differential expressions. The Vicanus plot is an effective visualization tool for differential expression research because it combines statistical significance with amount of strength, making it simple to find proteins with significant difference between experimental conditions. Like we are showing in color, the green and in the left side as the down-regulated protein. And on the other side, the red colorized are the upregulated. The gray point of proteins means no significant difference, and there are more far away of the point. So which are more far away, the more significant express is actually in the group. And then heat map. Heat map provides another visual representation of multi-dimensional data. displaying variables in a matrix format with colors and indicating relative values according to a scale. So the color of scale. This method efficiently reveals similarities and difference across multiple variables, such as the relative abundance or intensity of features in the MS data, making it useful tools for highlighting upregulated or downregulated patterns across the sample. Yeah, you can see normalized. Here we will have class of sample, and so each of the square here represents one set of sample. And maybe this is the groups, and this is another groups. And all this on the white axis, it represents maybe a protein or maybe a metabolite, or maybe even a transcriptome. It depends what you want to compare. And mainly with these two sets of map, we can distinguish these two differences between groups. Then we will reference the data bank, like function or pathways. So the bubble diagram can show the significantly changed metabolite pathways. With the pathway map, you can find the location of the relation of the metabolites or proteins and metabolites mainly. You can see also the relative metabolites and enzymes and so on. But it's still talking about too much tools. It's still too much script for most of the people to run through this data analyze. Don't worry, BGI Genomics provides a web-based solution for all this convenience analysis, visualization, and interpretation of data. Your omics data from RNAs, proteins, and metabolites can upload to the platform and finish all the analysis I just mentioned. In each of the map, like the volcano map, you can click to get information of the proteins you are visiting directly on the map. So only a few clicks without any scripts to be written, you can get a full idea of your data set in a few minutes. In total, you can simplify, send your samples and specific your analyzed data to us. The rest, it will take over and you can be more focused on your own research. The last of the section, I will be great to share some case studies to let you know more how to apply this to some research area. Again, single omics can only provide us an incomplete picture of the objects we are studying. A multi-omics can help us understand it as a full picture. Example, RNA and proteome is one of the most famous combination of the omics. We know that there are several steps between DNA expression to protein synthesis. And we even know that what is the code for mRNA to amino acid. But in many of the cases, they're not expressing together in a particular time. So there is no correlation between mRNA and proteins level in a single cell in a particular time. Yes, it may be measurement, noise, interfacing, or false positive, or it could be more complex machinimas in biological models. We don't know that. But combine this both two sets of data, researchers can use to examine multi-level regulation of gene expression and obtaining a panomics status of gene expressions. Correlation analysis panel at BGI Genomics providing is also in this purpose to discover a certain machinima pathway and elucidates a process. The different expressed gene and proteins will be analyzed separately. The correlation between them will be demonstrated in different methods and also functional enrichment, the network association, and transcription factor will be also analyzed. I'm taking an example of a single-cell RNA sequencing combined with single-cell proteomics. They first treated the mice with high-fat diet treatment, isolated islet cells as alpha, beta, and delta. The separately analyzed for these isolates can clearly explain the functional status in glucose interface in different isolates, such as this kind of ACE2 low alpha cells. They have significantly reduced metabolism and other biological activity, and they are less able to resist high fat diet stress. But the presenting in beta cells and delta cells and also different. Also, in different time points, they are having different profile. With this information, the functional status of each of them will be deeply elucidated. Another classic research topic I want to mention today is the relationship between intestinal flora and derived metabolites and human diseases, means metagenomics and metabolomics correlation analysis. It was found that in many kinds of disease, both intestinal flora and blood metabolites provide changes. It's already proved that metabolites generated from a flora in intestinal can be observed by the cells and enter the bloodstream, and they are directly or indirectly associated to self-health system and disease development. Simplify this research idea, the discovery and verification is the most preferred in these publications. The separated analysis of metabolomics and 16S on metagenome and find the relationship of them. Another validation set of animal experiments can be done. In one way, the facial microbiota or single microbiota transplantation or metabolite supplement or inhibitory or see if any changes in the disease development. An example of this is a research on type 2 diabetes. The medicine Barberina and probiotics have inhibited the supplied inference to specific type of flora, and they have found some short-chain fatty acids in blood also changing together with the flora composition, which explain the mechanisms of the treatment in type 2 diabetes. So all in all, we can see that how is the challenges now is solved, and we can see the huge potential for the future for multi-omics data set analyzing.